today's lecture, we're going to talk about DNA repair. I'd like to focus on three things today. So repair of mistakes that are made during DNA replication, and then also repair of DNA damage on a single strand of the double helix, and then finally looking at repair of strand on a double-stranded break. Okay, so an overview of DNA repair to show you some details here. So basically, repair of mistakes made during DNA replication is the definition of DNA repair. You can repair DNA damage on a single strand. You can also repair it on a double strand. I'm going to talk about the differences between the two. Before we talk about repairing DNA damage, let's talk how we detect DNA damage, or how we look for it, and specifically how we use something called the Ames test to look for potential chemicals that cause mutations and hence we believe to cause cancer. So it's a very interesting test that was developed in 1973, okay, so about 42 years ago, by a scientist by the name of Bruce Ames. And he had an assumption in this test. And the assumption that was that both cancer and mutations result from damage to DNA. Sort of an assumption. So there could be other things that, there could be variations to that, but it's an assumption that he made. Verification of his assumption is that over time we've realized that 90% of things that we know to cause cancer, right, so known carcinogens, are also mutagens. Then he used bacteria to show this, okay, sort of develop a test to detect for chemicals that are carcinogens. And just a quick side note, the bacteria he used were bacteria that were oxotrophic. They were bacteria that had defects in their liposaccharide coat, and they also had defects in their DNA repair mechanism. This is sort of the idea behind that. If we're looking for chemicals that may cause mutations to DNA and may be cancerous, we don't want these bacteria to be repairing their damage that's caused, right? Because it would be harder to detect if they have damage or not. So we want them to be just like a, like a fish in a barrel. Like you take a shot, you're going to hit it for sure. So we don't want things to be repaired. Also, we want their polysaccharide coat to be defective. If it's defective, that will make sure that the chemical is allowed to enter into the bacterium. So those are just a side note. He had to use those types of bacteria to assume that it was going to work. Okay, so the bacteria he used were also something called his-negative. Histidine is an amino acid that's required for bacteria to grow, but these bacteria had the inability to synthesize histidine from basic components. And it's very, very important. So put it this way. Here's his control group on the left. He took these his-negative bacteria, exposed them to something that was not a mutagen, right? So basically nothing, like water or something. He put them on a plate, but he put them on an agar plate that lacked histidine. If the bacteria can't produce histidine, the plate doesn't have histidine, and they need histidine to live, what would you expect on the control plate? You expect no growth, right? So no growth is our control. Now you might say, wait a second, there's a spot right there. And there is, but the diagram's just trying to show you that that's a reality, right? That things don't work perfectly. So expect no growth, but there might be something just to show you. That's all they're showing you. Okay, then on the right, what he did was this. He exposed bacteria, these his-negative bacteria, to a different chemical. If this chemical was a mutagen, that's what I want to focus on. So if it was a mutagen, and he put it on a plate that did not lack histidine, what probably would happen is if that chemical causes mutations in the bacteria by chance, and only by chance, right, some of the mutations would lie in the gene that allows the bacteria to produce histidine. Excuse me, let me say it again. Some of those mutations would lie in that gene that allows bacteria to produce histidine, and hence what would happen is it might enable the bacteria to produce histidine again. So mutations that are yielding a positive benefit. Now, there's no intention in that. I just want to mention that. So in other words, it's not like, they're intentionally, mutations are accumulating preferentially in a given area. That's not the case at all. It's just that some of the mutations might fall in that area, and some of them might enable the bacteria to produce histidine again, even though the plate here does not have histidine, right? So they could produce it from other components. If that happens, then you're going to have bacteria that are happy and can grow. So you could sort of envision that the more colonies you see here on this treatment plate, the stronger that the chemical's mutagenic properties were. So it's opposite of what you'd think if you just glanced at this experiment without looking at it in depth. In other words, if you saw, said which of these plates has the stronger mutagen, you would say, well, this one, or excuse me, this one does, right? There's no growth. But the way the experiment is set up, it's actually the opposite one, right? So this is the one that was exposed to the stronger mutagen. One other side note, you can envision a third group here. So let's say this plate didn't have any colonies or had like one. There's a third group he includes too, and this third group, what he does is he includes liver enzymes in the experiment. And the reason he does this is because you can envision that there's some chemicals that might not be mutagenic per se, but when the human body processes them, they turn them into mutagens. And so the liver enzymes are there to mimic that process. Okay, let's talk a little bit about once mutations occur, what happens. So DNA polymerase has the ability to actually proofread, right, to fix errors in DNA as it synthesizes DNA. And so every time the polymerase is adding a new nucleotide, it looks back after it added it to make sure that it's the correct letter. So if you think of my cursor as being DNA polymerase, it's moving around here, adding bases, adding bases. Before it goes to the next base, it's going to look back this way. It's going to say, did I add the right one? If not, it's going to do this. It's going to enact its 3' to 5' exonuclease activity. When you think about those words, right, think about the direction. Makes sense, right? It's backwards from replication because it's looking backwards. And then exonuclease, it's at the end of the strand because it's at the end that it's still synthesizing. It's going to enact its activity. It's going to chop off that improperly paired base or nucleotide, and then it's going to replace it with the correct one. Okay, quick question for you. So if you look at these plates and I said which of these was treated with the strongest carcinogen or strongest mutagen, which one would you say? Okay, think about it another second. Hopefully you're saying D. Remember, if there are no mutations, you're not going to get any growth, right? Or very little, perhaps. There's always errors. The stronger the mutagen, the more the bacteria you're going to see on the plate, the more colonies. Again, given the fact that he's using these histidine-negative bacteria, these his-negative, and the fact the plates don't have histidine. So it's all about the way the experiment's set up. Okay, about only 1 in 1,000 DNA lesions actually are maintained, right, so not repaired, to become a mutation. I'll tell you about a few statements about DNA repair. Usually requires 2 strands, right, 2 DNA strands, and it's redundant. What I mean by that is we're going to talk about 4 mechanisms of repair in a second here. 1, 2, 3, 4. And what you can see is the types of things they fix. You might see them in multiple categories. So in other words, if you look carefully through here, let's just see. Direct repair repairs pyrimidine dimers. And look, so does base excision repair. That also repairs pyrimidine dimers. So there's redundancy in the repair mechanisms. That's something that's sort of important. Okay, what I'd like to do now is let's look at that previous list, slide by slide, right? Repair mechanism by repair mechanism. Let's just talk about how it works. You get an idea, sort of a taste for how these things work. So the first type of repair I want to talk about is something called mismatch repair, and I'm going to show it to you in prokaryotic organisms. Whenever you have mismatch repair, we indicate a mismatch by a little bubble on one of the DNA strands. So here's the new DNA, here's the old DNA. The replication should have been what here? Should have been A, right? But it's G. So we bubble out the DNA to show a mismatch. Something that's interesting in bacteria is that eventually, when it's first made, the DNA is not methylated, the new strand. But eventually, DNA is methylated. So the old DNA templates that are used are always going to be methylated at these sequences called these C-tag sequences. Just put that in your memory bank. That's a way to differentiate old bacterial DNA from new bacterial DNA, the methylation of this C-tag sequence. Okay, so let's say there's a mismatch. What happens when there's a mismatch, a complex of enzymes called the mismatch repair complex is going to land on the mismatch. It's going to scaffold, there's a bunch of protein subunits, they're going to scaffold from the mismatch back in space, right? Back in this direction until they hit the first methylated C-tag sequence. When they hit that C-tag sequence, that's where the other part of the mismatch repair complex is going to anchor. The next step is this. Once it anchors across those areas, it's going to excise or remove the new strand all the way from the mismatch all the way back to the C-tag sequence right over here. Then what happens after that is DNA polymerase will come in, it'll fix the error, right? So it'll replicate that whole region there, and then DNA ligase will come in and seal the deal. You'll notice on these repair mechanisms, DNA ligase is always the last thing that comes in and seals the deal. It seals the phosphate backbone. You might look at this and say it's not very efficient. We had one little mistake, right? Just one little problem. We took out this whole segment of DNA. Remember, no one said nature's efficient. It's just the way that it evolved. Okay, what happens when there's a base that's damaged? Okay, a base is sort of a segue slide, so we won't talk about the diagram, but what happens when a base is damaged? Something called direct repair can happen, and what direct repair is when the base itself is fixed. So it's not removed, but like the errant functional group that was added is removed. So this is an example of this. So this base should be guanine, right? But when it was synthesized, it had this methyl group put on this oxygen when it shouldn't have been. And so instead of having guanine with this modified form, this methyl guanine, So what happens is, and this is pretty simple, this one actually, but an enzyme called methyltransferase comes in, removes the methyl group, and returns the base to its original form. Okay, and that's all there is to it for direct repair. Again, at the intro to genetics level, right? We could always probably teach a whole course on direct repair, but you know, at the basic level, this is how it works. There's another type of thing called base excision repair that removes and replaces a damaged base. This one's has a bit more steps to it. So what happens here is you have our old DNA strand on the bottom, the new strand on the top. There's a damaged base, right? There's a problem with that base. So what happens is an enzyme called DNA glycosylase comes in and removes the damaged base, takes it out. So what it leaves behind is something called an AP site. And this is a case where I think biologists are over naming things, in my opinion, but you know, we like to name things. What I mean by this is, what is an AP site? Well, what used to be here? Think of it that way. There was a base. Either it was a purine or it was a pyrimidine. One of the two, right? Now the purine or pyrimidine is not there. So AP means this, apurinic or apyrimidinic. In other words, a place where there used to be a purine or there used to be a pyrimidine. Either case, it's AP, right? Because of the way the letters work. And it's just saying it's not there anymore. Okay, so don't let that phrase throw you, right? That's all it's saying. Okay, then what happens is an AP endonuclease comes in and it removes the rest of the backbone in that region. Notice endonuclease. It's not chopping at the end of the strand, right in the middle it's cutting. Then what happens is DNA polymerase comes in. Again, the end of these is always so similar, right? The last few stages of all these repair mechanisms. DNA polymerase comes in, fixes the now nucleotide that was removed, and then DNA ligase comes in and seals the backbone, seals the deal. And that's the way that we repair these types of base excision repair, the damage to the base, according to this method. Okay, there's another thing called nucleotide excision repair that removes bulky lesions. So here what we're saying is we have damage to the DNA, perhaps a bit more substantial, maybe a few bases are damaged. And what happens is this complex of enzymes come in. I'm just showing it one big sphere here. But they come in and they land on that site where the DNA is damaged. They separate the strands, and then you might remember these cluster of grapes from the DNA replication lecture. These little clusters of grapes, right, there's protein complexes, they're called single-stranded binding proteins. And they keep the strands separated long enough for the complex of enzymes that originally landed to remove the segment that has the error. After that's removed, then again, polymerase comes in, re-replicates that segment, and then DNA ligase comes in and seals the backbone. It seals the deal. You might be noticing a lot of similarities between these. And remember, we said there's a lot of redundancy in DNA repair mechanisms. Okay, those were all with single-stranded breaks. Let's talk about what happens when you have a double-stranded break. This is much more problematic. So when you have a double-stranded break, you can't use the other strand, right? If this blue strand's broken and this one's broken, you can't use either of the blue ones to repair the break. So instead what happens then is you have to use the homologous chromosome, right? So if these two blue ones are a duplicated number 1 from the father, these two red ones would be a duplicated, what? Duplicated number 1 from the mother. So we have to use the homologous pair. So what happens is enzymes come in and they make a staggered break. So in other words, wherever there was the break to start with, they make it worse temporarily, and they make it staggered, right? They make it staggered break. Then what happens is one of these strands comes in on the broken chromosome, and it does something called strand invasion. Sounds like a Space Invader movie, but strand invasion. Come in, they invade the double helix, right? One of the strands pops out. And then what happens is a series of events where each of these red strands is used as a template for DNA polymerase to come in and resynthesize the broken strands. And that's what you're seeing here, right? And here. At the end of the day, what happens is something called holiday junctions are formed, where the DNA is sort of intermeshed or tangled together. Those are separated through a variety of mechanisms, which we're not going to cover in this lecture. So we'll just leave it at that holiday junction. But the main take-home point here is the fact that if there's a double-stranded break, you need homologous chromosomes to repair that break. Okay, so what happens when repairs are not made? So we spent the whole lecture today talking about single-stranded breaks, double-stranded breaks, the different repair mechanisms, the general qualities of DNA repair. But what if the DNA is not repaired? What happens? Well, if it's not repaired, that's most likely when we end up having issues with things like skin cancer or other types of cancer. Some people are more susceptible to skin cancer than others if they're fair complexion or if they have other genetic abnormalities. Basically, this is showing you skin cancer that's results from a defect in someone's nucleotide repair mechanism, nucleotide excision repair mechanism. And this is, overexposure to UV. A normal individual could handle this level of exposure, but if they have defects in these repair mechanisms, a person is more susceptible, and that's the types of skin cancer that you see here.